Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about non-selectable marker gene or reporter gene. Non-selectable marker genes or reporter genes. The genes are encoded with visually identifiable characteristics. Reporter genes are also called as visible marker genes because they change the appearance of the plant tissue. So reporter genes are used to tag another gene or a specific DNA sequence, for example promoter. Reporter genes are useful to quantify gene expression. Reporter genes can also be used to study the effect of transcription factor on gene expression. The genes can also be used to study the function of various gene regulatory elements. In addition to this, reporter genes are vital in identification of tissue-specific and pathway-specific gene expression. In this example, this is the reporter gene. This reporter gene is fused with the regulatory sequence. This is the regulatory sequence that belongs to another gene. The regulatory sequence can be a promoter sequence or any other sequence. So, by fusing reporter gene with a gene regulatory sequence, we can study the function of this regulatory sequence. So reporter genes are very much useful to study the function of various gene regulatory elements. So reporter genes are transcribed that result in the formation of mRNA, and mRNA is later translated to form reporter protein, for example GFP. So the amount of protein that is formed can easily measured through various assays. So by using reporter gene, we can easily study the function of various gene regulatory elements. Several reporter genes have been described in the scientific literature, but three of them are more popular. These three systems are UIDA gene. UIDA gene, also known as Gus gene, the gene system code for the enzyme beta-glucuronidase. Another reporter gene is LUG gene. LUG gene code for the enzyme luciferase. And the third example is GFP gene. GFP gene code for a protein known as green fluorescent protein. Out of these three reporter genes, UIDA gene and LUG gene are example of conditional non-selectable marker gene because they require the use of an external substrate for detection of activity, whereas GFP gene is an example of non-conditional non-selectable marker gene because the protein that is encoded by the gene is directly detectable. First of all, we shall discuss about UIDA gene system. UIDA gene system is also known as Gus gene system. This gene is obtained from E. coli and this gene code for the enzyme beta-glucuronidase. UIDA gene system is the most widely used reporter gene in plants. This Gus gene system is useful to study the function of various gene regulatory elements and mechanisms. It can also be used in promoter trapping experiment. Several Gus gene assays are available depending on the substrate that is used. First assay is histochemical staining. For Gus histochemical staining, The substrate is 5-bromo-4-chloro-3-indolyl glucuronide, which is abbreviated as X-gluc. So the substrate is hydrolyzed by the enzyme beta-glucuronidase to form a product that is known as 5,5-dibromo-4,4-dichloroindigo. This compound is abbreviated as DIX indigo, which is blue color. So the development of blue color due to this hydrolysis can be observed by microscope. The activity of GUS-A gene can also be studied by spectrophotometric assay. For spectrophotometric assay, the substrate is p-nitrophenyl beta-D-glucuronide. Similarly, GUS-A gene activity can also be studied by fluorimetric assay. For fluorimetric assay, the substrate is 4-methyl-umbelliferyl beta-D-glucuronide. So this is about UIDA or GUS-A gene system. GUS-A gene system is the most widely used reporter gene in plants. Another example of reporter gene is lock gene. Lock gene is obtained from the firefly known as Photinospiralis. The lock gene code for the enzyme known as luciferase. The enzyme luciferase catalyzes the ATP-dependent oxidative decarboxylation of luciferin. So, luciferin is the external substrate that has to be added to the selection medium for detection of activity. And this oxidative decarboxylation of luciferin emit light, which is an example of bioluminescence. This lock gene system is a sensitive, non-destructive monitoring system that allow real-time analysis. However, this reporter gene has not been used as extensively as the GUS gene system. Another important reporter gene is GFP gene. The GFP gene code for a protein known as green fluorescent protein. The green fluorescent protein exhibit bright green fluorescence when exposed to light in the range of blue to ultraviolet. The GFP gene is an example of non-conditional, non-selectable marker gene because there is no need of adding any external substrate to the selection medium in order to detect activity. The green fluorescent protein traditionally refers to the protein that was first isolated from the Pacific jellyfish known as Euphoria victoria. And the green fluorescent protein that was isolated from the Pacific jellyfish is known as AVGFP. The GFP gene is used to tag various plant genes in order to understand the protein interaction and protein trafficking. The GFP gene system is also useful for easy detection of transgenic plant and plant parts such as pollen. GFP gene is also useful in modified form as biosensor. The GFP gene is not only present in the Pacific jellyfish but also is present in various animals like corals, copepods, zoanthids, sea anemones and lancelets. Friends, this is about various reporter genes like GUS gene, lock gene and GFP gene. The GUS gene and lock gene are example of conditional non-selectable marker gene or reporter gene whereas GFP is an example of non-conditional, non-selectable marker gene or reporter gene. Thank you friends. Please share, like and subscribe this video, and also please give your comments so that I will be able to improve my performance. See you. Bye bye.